Oh, okay, let us now introduce our first speaker. Özlem Türeci is a physician by training, an academic researcher, and she's also an entrepreneur. Türeci and her partner, Dr. Ugan Sahin, were pioneers in developing the messenger RNA vaccines. They improved their potency and they found a way to use them successfully in humans. In addition to being a key person in the race for the COVID-19 vaccine, Özlem Türeci is a co-founder and director of BioNTech, which is a company where she is in charge of the clinical development of a number of different oncological approaches, and she also supervises more than 30 international clinical trials. She's also received dozens of prestigious awards for her work and holds more than 500 patents. The title of her lecture is Molecular Communication with the Immune System. So, a warm round of applause, please. Thank you.
From the DIPC, dearest Pedro Miguel, and dear ladies and gentlemen, it's a pleasure to be here in this wonderful Basque country, in this vibrant town, and here in this marvelous theater. So thank you very much for having me and for inviting me. We scientists, ladies and gentlemen, are driven by two forces. One is the passion for knowledge, and you know that, and the second is that we feel the calling to contribute to a greater cause, even if it's just a tiny bit which we can contribute. And I would like to start my talk by sharing with you the greater cause which inspired us to get interested in molecular communication with the immune system. Many years ago, we were young medical doctors taking care of cancer patients, and more often than not, we had to deliver a devastating message to our cancer patients, namely that there was no medicine against their disease left after they had not responded to the standard of care. And you can imagine that we could only endure this crushing, almost daily experience because we were leading a double life. By day, we were at the bedside of our patients, and in the evenings, in the nights. Is this better? Is this better? No? Is it better if I stand here? Okay. So in the evening and in the nights, we were at the lab bench and were going down the rabbit hole of science. And there were actually two research fields we were interested in. One research field were cancer cells, which we studied, and we understood that these were cells gone wild. Cancer cells evolve from normal cells by accumulating over time random aberrations in their genetic, in their DNA, in their genetic information. And these aberrations are random. And this is the reason why each and every cancer is as unique as the afflicted person. And this made directly clear why the one-fits-all medicines which we were offering our patients would not help. The other system we were interested in, or the other research area, were the forces of the immune system. The immune system which has evolved over a long time for high precision fighting and defense of enemies. The immune system consists of many different forces which are specialized. One of those which I like to imagine, yes, like the ninjas shown here, are the so-called killer cells or T cells, and they are particularly potent in killing enemy cells. The drawback is that the immune system has evolved and is specialized to defend against external pathogens, but not internal aberrations. We, however, ask the question, is it feasible to reprogram the immune system such that it goes against the cancer of our individual patients? And to do this, you need communication. You have to communicate with the immune system. And the way this communication goes is what we call vaccination, working with a vaccine. A vaccine is like a wanted poster, where you tell the immune system about the enemy. And three things are important if you work with such a wanted poster. You have to be very precise. So you need a format, a tool which helps you to very precisely describe how the perpetrator looks like, so that there are no mix-ups. And the second is that you have to be very potent. Potency is very important here. So that means, again, that the wanted poster has to be distributed widely so that you have many forces on their toes to find the enemy, the perpetrator, and act against it. And third, you need clear language, clear instructions. What should the immune systems do when finding this perpetrator, which in this case means attacking? Back in those days, and these were in the late 1990s, all the different technologies we looked into, which qualified or were used as vaccine technologies, did not appear to fulfill these requirements to good communication tool with the immune system, until we stumbled over messenger RNA, mRNA. mRNA is the basically oldest information technology invented by nature. It has been discovered in the 1961s. In 1961, there were two publications back-to-back, actually, which were authored by 9 scientists, and 3 of them are shown here, which described the discovery of this molecule. And shortly after that, there was a paper by François Jacot, who is also on this slide, and Jacques Monod, who described the principle, how this newly discovered molecule played a role in very fundamental processes in cells of organisms. The way mRNA works naturally in almost all of our cells, and also cells of organisms, other organisms, is shown here. Our genetic information, the so-called DNA, so the hardwired information, is within a special compartment. Can you see the, yeah. Within a special compartment of the cell, which is called nucleus, where it is protected. And it does not come out of there, it just lies there. And there is a process which is called transcription, by which blueprints of this information are generated on demand. And this is what we call mRNA. mRNA goes out of this valve, out of this protection part of the cell, into organelles which we call ribosomes. And these are the protein production factories of the cell. And it's like a conveyor belt in which mRNA serves as a template for the information which needs to be transmitted and which needs to be translated, this is how we call this part of the process, to protein. And the proteins, the billions of proteins in the cells, are those elements which coordinate life, which build up the cells, and which are basically the important molecules. And after mRNA has done its job, it disappears. You have to imagine this like you have a cookbook, which is the DNA, and you jot down a recipe to take it to your working place in the kitchen, which is the mRNA, and the cake or the souffle which you are producing is the protein, and you throw away your piece of paper, the mRNA, in which you have written down the transient information. So the way how mRNA translates is very well defined. So there is a 4-letter code, and there are very clear rules how what is written down in the DNA is translated into mRNA and is translated then is then translated into protein. So this information technology is very precise and can describe exactly what you want to describe. And we ask the question, why can't we use mRNA as a way to communicate with the immune system? Can we use mRNA to convey the wanted poster of the perpetrator of the cancer cell? And how can we do it? The interesting thing for us was that mRNA is not the wanted poster itself. It's the blueprint. So bringing in a blueprint into a patient who then uses this blueprint to print many, many times this wanted poster, which this appeared like a very attractive way to go. We saw the potential of mRNA as a vaccine, but we also understood that there was a long way to go, many improvements needed. One of those drawbacks back in the 1990s, why mRNA was overlooked by many scientists, was that it had a very short half-life. So the information was there for a short time, which meant that even high doses of mRNA ended up in little amounts of protein and therefore little amounts of effect. And we looked for ways to optimize that, to optimize the performance of the mRNA. Now, mRNA has different elements. This is such a strand of mRNA, and there is one part which is the blueprint of the wanted poster, which in the cells, in the protein production facilities or factories, can be used as a blueprint to produce the wanted poster, but there are also multiple other elements as shown here. And we found out that modifying each of them by molecular engineering ended up in prolonging the presence of mRNA in the cells of the patient and optimizing the amounts of protein and thereby the potency we could achieve. And combining of all of those brought us an increase of several orders of magnitudes of the effect. And you can see this here, that the wanted poster was all of a sudden expressed for a much longer time. So that meant by molecular engineering of these parts, we were endowing the mRNA with a VIP pass at the protein production factories. Our colleagues, Drew Weissman and Kati Carico, used another path to come to the same effect. They modified, and this is represented here by these symbols, they modified the chemistry of the mRNA and thereby blunted its recognition by defense elements of cells in which we bring in the mRNA. That means they endowed the mRNA with a stealth mode in order to ensure more protein and more effect. In the COVID-19 vaccine, both these elements, molecular design and the chemistry, are combined. In our cancer vaccines, we only use our approach because we need a more stronger immune response. So this was one field in which we ensured improvements, which was a breakthrough because all of a sudden it increased the potency of the mRNA to induce induce a strong, precise immune response. There was another area in which improvements were needed. When you use a vaccine, you want to communicate with the immune system. And you have to bring the vaccine to the place where this communication makes sense and has the largest effect. In the body, there are specific organs which are called lymphoid compartments. The spleen is one of them. The spleen is one of them, and all the lymph nodes, which are all across your body, are other parts of this large disseminated organ. And these are the places which you can imagine as boot camps of the immune system. Also physiological immune reactions, communications with the immune system, take place there in these organs. And in these organs, you have different cell types of the immune system. However, there is one cell type which is called dendritic cells. And this specific cell type, these are the coaches, the high performance coaches of the immune system. So what you want to do is to get the information, the blueprint of the wanted poster, to these generals who are meant to train the army of your killer immune cells. And so another approach or improvement we ensured was that we developed nanoparticles which, when given intravenously into the bloodstream, traveled in a targeted way to these organs, to lymphoid compartments, and in these organs reached the dendritic cells, the specific generals or trainers of the immune system, and were then used by those to copy, to print the wanted poster and to train basically body-wide those killer cells which were needed for the attack. And you can see what that exactly means here. The blue ones, I'm not sure whether you can see this in the slide. The blue cells are cancer cells, and these cells here are the killer cells of the immune system. They are trained to find the right cancer cell, to stick to it, and to kill it with what we as immunologists call the deadly kiss. One important piece with both improvements was that we could get up the potency so that little amounts of mRNA were sufficient to get a very strong immune response. And as compared with the mRNA formats with which we started, these improvements ensured a more than 1,000 times higher potency. What did this mean? In the COVID-19 pandemic, this meant that we were able to serve a large part of the global human community, because we were able to have a potency increased such that this small bag was sufficient to get 8 million doses of our vaccine. So potency, in particular in global crises such as pandemics, is a very important piece. So, as you might remember, when I started, I talked about cancer and how unique cancers are, and that basically each and every cancer patient has a unique and individual cancer. One of our dreams was to use mRNA, which is a very versatile and easy to manufacture technology, to use it for individualized vaccines. So imagine you have the cancer of one patient, you analyze it, and for this there are technologies which are called next generation sequencing, which give you a high resolution of all the aberrations which are in this cancer. And you find all these aberrations, you use artificial intelligence, AI, in order to select those aberrations which precisely and potently describe this cancer. And then you use mRNA in order to, on demand, in a tailored way, manufacture a vaccine for this very patient. We started with clinical trials doing exactly that. So on-demand manufacturing for individual cancer patients in 2014, and each vaccine took us long. For each and every patient, we needed 3 to 5 months to deliver the vaccine. We treated hundreds of patients over the following years until 2019, and we improved the technologies such that we only needed 3 to 5 weeks to deliver the personalized vaccine in clinical trials which were conducted all over the world. And you can, by knowing this, understand why we were able to deliver a COVID-19 vaccine so fast in only 10 months, because we could build on this expertise which we gained running the production race for each of our hundreds of patients over and over again, each time a race against that patient's growing tumor. And this might also help you to understand why we are able to adapt the vaccine now for each new strain which appears in each new season, as also in this season. That meant that at least in clinical trials, we could move a bit closer to our dream that we were not able to provide standard of care for the cancer diseases of our patients, but we could build them their individualized vaccine. And we hope that one day this will also become, after it has gone through clinical testing, can become a standard of care. So mRNA is an entire toolbox. In the meantime, over the many years now, in which generations of scientists have first contributed to understand mRNA as a biological molecule, how it is regulated, how it works physiologically in cells. Generations of scientists also have contributed to understand how you can tweak the chemistry, the format of mRNA, and also the formulations, the nanoparticulate formulations, to use it preventively and therapeutically in humans, in clinical trials, but also as an approved product. This all has generated an mRNA toolbox in which we have basically different formats of mRNA, also mRNA which comes with its own turbo, which can basically in the patient also amplify the blueprint itself, which is self-amplifying and trans-amplifying mRNA. And we have different types of nanoparticles in which we can envelope the mRNA, which ensure that we can send mRNA to different places, not only into lymphoid compartments, but different places like the liver or other organs where for some types of diseases you need the targeting. And we can encode different types of molecules depending on the type of disease. That means that mRNA has access to many fields now in which it is tested. Cancer is the field which I have already described to you, where multiple clinical trials in advanced stages are being conducted worldwide. Infectious diseases, yes, COVID-19 vaccine development has been successful, but we are also going now into other infectious diseases, including those which are a global health burden, like malaria or tuberculosis. And then there is autoimmunity. You can use mRNA to tell the immune system to attack a certain cell. This is what we do, for example, in cancer. But you can also modify the instructions such that you tell the immune system to hold back. And this is what you need in autoimmunity, where the immune system attacks your structures, your bodily structures, for example, in multiple sclerosis or rheumatoid arthritis. And then another interesting field in which we think mRNA will transform is protein replacement. Diseases where a certain protein genetically is deficient or does not work and causes the diseases, here we can replace with mRNA. With this, I would like to end, and thank you for your interest. And this last slide, actually, is for the young ones here in the audience. I have seen that we have many young, either scientists or young people who are interested in science. And to you, I would like to convey the message: many things in science which become breakthroughs start tiny, but can become powerful. So I would like to encourage you to go into science, and if you are there, to be persistent and follow the path. Thank you very much.
Thank you very much.
Sí.